Welcome to the last part of the talk, Key Space Trajectory Imaging. In this part, I will be talking about a novel framework to resolve a kurtosis source in a more general way using the correlation tensor imaging. So just a quick recap of the last talks. So with key trajectory encoding, what we basically use is we assume that the tissue can be represented by a sum of multiple Gaussian components. And based on this assumption, different key trajectory encodings can just be characterized by its B tensor. And this information can be useful to decouple the total kurtosis source into two different sources. The anisotropic kurtosis sources, which reflects the microscopic anisotropy of a system, and the isotropic kurtosis, which reflects the variance of the sizes of the different components. A limitation of this B-tensor framework is that it assumes that tissue can be represented by the multiple Gaussian approximation. However, tissues can be more complex for this assumption to be valid. So with the correlation tensor imaging, our objective is to provide a more complete picture of the tissues. So now we divide the tissue and components, and each component will not be only classified by apparent diffusivity, but also apparent microscopic kurtosis. And each component will have will be characterized by their own individual diffusivities and individual microscopic kurtosis. So what can be this microscopic kurtosis? So as I explained on the first DKI plot, individual compartments can exhibit non-Gaussian properties due to the confinement of diffusion inside different compartments. So microscopic kurtosis can rise due to the interactions of the diffusion molecules with the boundaries of the compartments. Also, tissues can present complex features. So each compartment can show features that doesn't fit well with a Gaussian component, as for example, a cross-sectional variance along axons. And this was shown on previous work that it produces an effective microscopic kurtosis. So microscopic kurtosis can also rise from these effects as cross-sectional variance. Another effect is exchange. So if we have exchange between two compartments, even if the diffusivities are similar, this can rise a non-Gaussian component that will reflect a microscopic kurtosis term. Of course, that in real tissues, all these effects can be happening simultaneously. So what we measured at the end is a total effect of this microscopic kurtosis, which is basically a weighted average of all the individual microscopic kurtosis terms. So if you got lost with all this information, the main idea here with the correlation tensor, so the home message here is that we are trying to develop a strategy that will decouple kurtosis sources not on only two sources related to variances, but in three sources. So we will still estimate K aniso and K iso, but then we will have an extra term that will estimate any deviation from this Gaussian picture. So then after we develop the strategy, we also want to apply the CTI and show the potential on providing more complete characterization of microstructural alteration in both health and disease. So for example, in aging, stroke, or in brain tumors. Just to give you an example of CTI potential. So for example, in stroke, it is known that DTI and DKI metrics are already very sensitive to early microstructure changes of ischemic lesions. This early sensitivity is essential for stroke treatment. However, improving stroke imaging is still essential to improve the criteria for the inclusion or exclusion of subjects to different treatments. CTI has a huge potential to improve the specificity of diffusion MRI because it can target different biological mechanisms. So, for example, mechanism has a presence of edema on the stroke lesions, or effects of swelling or beading, which basically are change on the microscopic morphologies of cells, so as shown here in this histological images. And these two different mechanisms will be targeted by different kurtosis sources. So, for example, edema, the presence of edema will lead to increase of K-iso, while increase of beading will lead to increase of microscopic kurtosis. And note that this microscopic kurtosis can only be measured by accurately by CTI and not by other key trajectory imaging techniques. So how the CTI works? So basically this technique uses the information of double diffusion encoding. So as explained on previous talks, the diffusion encoding sequence is a sequence that contains two diffusion encoding blocks. Each block can be characterized by unique direction and an independent magnitude. CTI is based on the signal cumulant expansion. So basically, it is a generalization of the diffusion kurtosis imaging. So for example, if we only consider the first diffusion encoding block, the CTI expression will basically match the standard diffusional kurtosis imaging, in which we can characterize the diffusion tensor and the kurtosis tensor for a given diffusion time, which is defined by the distance between the two diffusion gradient pulses of a single diffusion encoding. So now considering the two diffusion blocks, we can see that the CTI model now have more tensors. So which are these tensors? So we will have still this diffusion tensor, then we will have also an extra tensor that will modulate time correlations between the two diffusion blocks on the diffusion tensor. Then we will have the standard kurtosis tensor, Z tensor that, as I will explain, it is related to the covariance tensor, and AS tensor that modulates time correlations on the covariant tensor. First thing that we do in CTI is that we use the long mixing time regime. Basically, this means that the two diffusion encoding blocks have to be separated by a long enough time. And we do this basically to subtract time dependent correlation. So basically canceling the two of the tensors, so the Q and the S tensor, and also to be able to define the diffusion, the kurtosis, and the C tensor for a given time. In practice, we observe that the long mixing time regime is met when this time is as long as the big delta. So after removing the time-dependent effects, we can now focus on the three tensors, the diffusion, kurtosis, and Z tensor. And at the long mixing time, we also showed that this converges to the covariant tensor. And as I explained on my previous talks, the covariant tensor can be used to extract sources of kurtosis variance. So basically extracting K aniso and K iso. So the main idea with the CTI is first to compute the total kurtosis from the kurtosis tensor, a similar way that we do with diffusion kurtosis imaging. And then we extract K aniso and K iso from the covariant tensor. And after having these measurements, the microscopic kurtosis can be estimated as the difference between the total, the K iso, and the K aniso source. In the first proof of concept CTI study, we used it to resolve the kurtosis sources of ex vivo mouse brain using a 16.4 Tesla preclinical scanner. Basically, we used a large number of DDE B-value pairs combinations, as well as a large number of pairs of DDE gradient directions. Here I'm showing the total kurtosis source extracted by CTI, and here its sources. So we can see that the sources of kurtosis resolved by CTI predicts the higher K-ISO on the white matter of the rodent. We show that K-ISO is sensitive to the partial volume effect, so where we have higher variance between free water and the water in tissues. And more interestingly, we observe that the microscopic kurtosis is non-vanished, so it's present. A pitfall of this study is that the data was acquired with long acquisition times that were not compatible for in vivo acquisition. And also, since we did ex vivo acquisitions, this microscopic kurtosis, we don't know if it can be caused by fixation effects. In the following web study, we then focus on trying to minimize the protocol for CTI, basically to translate it to in vivo acquisitions. So what we found is that to reconstruct CTI, we need at least 4 sets of different diffusionmri acquisitions. So this includes 2 sets of parallel double diffusion encoding for 2 different b-value magnitudes. We also need another set of double diffusion encoding in which the 2 pairs of double diffusion encoding is acquired perpendicular to each other. And also we need a set that is equivalent to single diffusion encoding. We also found that each combination of these sets provide specificity to different kurtosis sources. Based on this, we can also show why resolving microscopic kurtosis is only possible with CTI and not using the B tensor encoding of key trajectory imaging. Because if we plot the effective B tensor values, we see that the 2 sets that are responsible to microscopic kurtosis have the same information on the B tensor. So there's no way using this framework to distinguish the information from these 2. We used the minimal CTI protocol on in vivo rodent brains. So this was done basically on 3 different rats that were under anesthesia using isoflurane, and the acquisition was done on the 9.4 Tesla glucose scanner with a rat cryoprobe. So here are the b-values and diffusion parameters that we use for these acquisitions. So here's an illustration of the double diffusion encoding timings that we used. Acquisitions took around 45 minutes in total. And here in the right, I'm showing just the powder average data of each of the sets of the minimal protocol, just to show you the quality of our raw data. The CTI results in vivo confirmed our previous ex vivo findings. So, for example, here is a total kurtosis. We observe that the K and ISO has the higher sources in white matter. Again, K ISO shows the higher intensities on regions near the ventricles. And we again observe that microscopic kurtosis is a predominant source of kurtosis. So it consists around 30% of the total kurtosis in white matter and 60% on gray matter. After this, we were ready to move to the stroke application. So this part of the project was led by a PhD student on the Chamesh lab, Rita Alves. So basically what she did, she scanned stroked animals 3 hours post-stroke onset using the Rose-Bengal mouse stroke model. And she performed both acquisition on ex vivo, just to have data with higher quality, and then she also applied this on in vivo animals just to show if these results were consistent on both ex vivo and in vivo acquisitions. Here I'm showing the standard mean diffusivity, FA, and total kurtosis of one of the ex vivo brains. As expected, the stroke lesions are associated with unspecific decrease on mid-diffusivity and increase of total kurtosis. Our kurtosis sources shows that the microscopic kurtosis is the leading source responsible for the increase of non-Gaussian diffusion in stroke, and this is in line with our expectations with the beading effects. The results were consistent across different animals and also consistent to our in vivo acquisitions. Confirm that beading can indeed be the effect that is increasing our microscopic kurtosis sources. We also did some complementary simulations of beading geometries, and this was done using the open source Python package dpy that was developed by Levy. And this confirmed that as we increase beading, we have an increase of total kurtosis that can be explained by increase of microscopic kurtosis. We are also currently working on the translation of CTI to a clinical scanner. So this is work done in collaboration with the University of Trento. And the objective here is to use CTI to characterize the kurtosis sources of healthy human brains. So for this, we used the minimal protocol of double diffusion encoding sequences implemented on a 3 Tesla Siemens scanner. Data was acquired for 10 healthy participants. The minimal protocol was acquired for a larger number of directions that we use on the preclinical study and a larger number of averages. So this is to ensure the good quality of the first CTI maps on humans. We also had to adjust the diffusivity timings of our sequence to the clinical scanner. So here is a comparison of a schematic of what we are using and what we use on the preclinical. And so this is on the same scale. And basically we are using higher diffusion times and higher small delta, and this might decrease a bit our microscopic kurtosis. However, looking to the maps, so this is the maps across the 10 different subjects. This is for the CTI mean diffusivity, CTI total kurtosis, K-aniso, K-iso, and microscopic kurtosis. So we can see that the results show consistent maps across different subjects. We show again that K-aniso has higher values in white matter, and that K-iso now shows higher intensities than the rodent data, because in human data we have a larger amount of partial volume effect between tissue and CSF. So this is confirmed on these maps. The microscopic kurtosis, although they show smaller values than our preclinical data, we still see that on the human clinical scanner conditions, it's still non-vanishing. So even that we had to increase our diffusion times in the small deltas. So we also registered all the data to a common template. So here is the templates of the different kurtosis sources of all subjects average. Again, we can confirm that we have non-vanishing microscopic kurtosis values, and so it's not a non-vanishing source. Looking to the percentage maps, we now see that across different regions, the microscopic kurtosis correspond around 20% of the total kurtosis, 15% of the total kurtosis, which is much lower than the data that we observe on the rodents. Despite this study provides very nice templates to start exploring some regional difference of the kurtosis source across different brain areas. So for example, here the microscopic kurtosis map. So here I'm showing different axial slices of the templates, and you can see that for the microscopic kurtosis, we have higher values in the prefrontal region, and this is in line with the higher total kurtosis values on this area that was reported on early DKI studies. In white matter, typically microstructure modeling assumes that there is no microscopic kurtosis on white matter, and we see that microscopic kurtosis is 0 in some of the tracts. So, for example, the lateral projection of the corpus callosum and in the internal capsule. However, this doesn't seem true to the generality of white matter. So this suggests that we might visit microstructural modeling in white matter, particularly when we assume that microscopic kurtosis is 0. Okay, so to summarize this first talk of the key trajectory imaging, the conclusions or the take-home message is that advanced diffusion encoding sequence is sensitive to unique information that cannot be resolved with conventional encoding. So, for example, during the talk, I showed that QTI and CTI can be useful to resolve sources of kurtosis that single diffusion encodings cannot resolve. Also, I showed the potentials of this technique in different applications. So I discussed the advantage on tumors on the first part of the talk, and now I showed the example of CTI and to characterize ischemic stroke lesions. It's also important to note that this can have general applicability to other applications. So, for example, in this study, double diffusion encoding was used to characterize lesions of multiple sclerosis, and then QTI was also used to characterize changes of aging and Parkinson disease. And for this, there's this very recent and interesting paper that just came out 2 years ago. So just some future vistas on QTI Dipy implementations. So there's a lot of room to improve QTI reconstruction models. So for example, adding positive constraints in the tensor. And on the next talk, we will hear more about this. I also intend to continue with the CTI translation to clinics. So try to further accelerate the CTI acquisitions to shorter acquisition times than 45 minutes. And then after that, I want also to make the CTI code available in Dipy. Then I think another interesting thing that we should do is to use CTI to try to assess the impacts of ignoring microscopic kurtosis in QTI frameworks. And this can be very, very useful. Why? Because if we can calibrate the QTI measurements to be insensitive to microscopic kurtosis, or even to try to incorporate it, this could provide a much faster strategy than CTI. So there's also some future work to do on this. So for further information on CTI, so I'm here showing the current manuscripts that were done related to CTI. Finally, I want to thank all my collaborators that make this project possible. And I want to thank you all for your attention. Bye.